Hello everyone and welcome to Biology with Dr. Soniyaa Shrivastava. So far, we have understood how photosynthesis takes place in plants. But is photosynthesis always efficient? Not necessarily. Under certain conditions, plants undergo photorespiration, a process that reduces the efficiency of photosynthesis. Also, the rate of photosynthesis is influenced by several external factors. So, in this video, we will study the photorespiration and the two factors which affect the rate of photosynthesis is light and carbon dioxide. So, let's begin. Let's start with our first MCQ. In C3 plant, some O2 binds to Rubisco, causing a characteristically wasteful process. What happens to the primary acceptor molecule RUBP during this specific pathway? Student, in the previous video, we have already learnt about the Rubisco and the wasteful process photorespiration. So before coming to the options, let's once again see those processes. We know that Rubisco is ribulose 1,5-bisphosphate, carboxylase and oxygenase. Student, we know that Rubisco has affinity for both, that is carbon dioxide and for oxygen. When CO2 is available, Rubisco acts as a carboxylase, and when O2 is available, it acts as a oxygenase. In case of normal photosynthesis, when CO2 is available, RUBP, which is having 5 carbon, reacts with CO2 having 1 carbon in presence of enzyme Rubisco to form initially the 6 carbon compound, which is unstable, so directly splits into 2 molecules of 3-PGA, which is stable molecule of phosphoglyceric acid. But when Rubisco binds to O2, photorespiration occurs. How? That RUBP binds with O2 in presence of Rubisco to form 1 molecule of 3-PGA, 3 carbon, plus 1 molecule of phosphoglycolate having 2 carbon. This is the waste or toxic byproduct. So when Rubisco binds with O2, it behaves as an oxygenase and photorespiration starts, a pathway that wastes energy and reduces productivity. Let's see our options. Option number 1. It is completely oxidized into 2 molecules of 3-PGA. This is the normal carboxylation pathway where RUBP combines with CO2. But in case of photorespiration, RUBP combines with O2 to form the waste product, that is phosphoglycolate. Therefore, this option is wrong. Option number 2. It is converted into 1 molecule of 3-PGA and 1 molecule of phosphoglycolate. Yes, this is our correct option. We have already seen that when RUBP combines with O2 through oxygenase activity of Rubisco, it forms the 1 molecule of 3-PGA and 1 molecule of phosphoglycolate. Option number 3. It splits into 2 molecules of oxaloacetic acid. OAA is the stable product of C4 pathway and is formed by PP carboxylase, not by Rubisco oxygenation. Therefore, this is also the wrong option. Option number 4. It binds with ATP to directly synthesize a molecule of sucrose. Photorespiration does not synthesize sugars. In fact, it consumes energy and reduces photosynthetic efficiency. Therefore, this is also the wrong option. Moving on to MCQ number 2. Which of the following statement precisely describes the energetic cost and metabolic output of photorespiratory pathway? Student, photorespiration is often called a wasteful pathway because the plant spends energy and releases previously fixed carbon as CO2 and produces no sugar. Let's check the metabolic profit and loss statement before coming to the options. In case of photorespiration, what happens? When O2 combines with RUBP, it spends energy in the form of ATP. So, because of that, CO2 is released. So, no ATP is formed, no NADPH and no sugars. So, let's come to our options. Option number 1. It synthesizes ATP and NADPH while releasing CO2. Photorespiration does not synthesize ATP or NADPH. Instead, ATP is consumed during this pathway. Therefore, this is the wrong option. Option number 2. It utilizes ATP and releases CO2 but does not synthesize sugar. Yes, this is our correct answer. Photorespiration results in the release of CO2 with the utilization of ATP. So, no ATP, no NADPH and no sugars. Option number 3. It consumes sugars to generate a high protein gradient in grana. Proton gradients are generated during the light reaction. Photorespiration occurs across chloroplast and has no role in building a proton gradient. Therefore, this option is wrong. Option number 4. It fixes CO2 without utilizing any metabolic energy tokens. Photorespiration releases CO2 rather than fixing it. Moreover, ATP is consumed during this process. Therefore, this is the wrong option. Moving on to MCQ number 3. This is an assertion reason based question. What is assertion given here? That photorespiration does not occur in C4 plants. What is reason given here? C4 plants have a special mechanism that completely increases the concentration of CO2 at the Rubisco enzyme site. Let us now see, is our assertion reason are true, and most important, is there any connection between them? So before coming to the option, let us once again see the C4 pathway. In C4 pathway, first the reaction starts in mesophyll cell, which is the outer layer of the leaf, where atmospheric carbon dioxide is captured by PEP, which is the primary acceptor of CO2. So, in presence of PEP carboxylase, OAA is formed. OAA is 4-carbon acid. So, as it is highly reactive, it gets converted into malate. Now, this malate gets transported to bundle sheath cells, which is the inner layer of the leaf, where decarboxylation takes place, and malate gets broken down into 1 molecule of CO2 and 1 molecule of pyruvate. This CO2 gets released into the location where Rubisco enzyme is present. Due to high concentration of CO2, Rubisco acts as a carboxylase. So, Rubisco binds with CO2. So, Calvin cycle started and sugars are then made. Now, let us see our assertion. What is assertion given here? That photorespiration does not occur in C4 plants. Is this statement is true? Yes, this statement is 100% true. Now, what is the reason given here? That C4 plants have spatial mechanism that completely increases concentration of CO2 at Rubisco enzyme site. Is this statement is correct? Yes, this statement is also 100% correct. We have already seen the C4 pathway that due to higher concentration of CO2, Rubisco act as a carboxylase and binds with CO2, and because of the low concentration of O2, it doesn't bind with O2, so photorespiration is prevented. Because photorespiration only occurs when Rubisco binds with O2. So reason completely explains the assertion. So let us see our options. As we already know that our assertion is true and our reason is also true, and reason completely explains the assertion. So our correct answer is both A and R are true, and R is the correct explanation of A. Let us see MCQ number 4. The dual nature of enzyme Rubisco allows it to bind with both CO2 and O2. What primary factors determines which of these two gases will bind to the enzyme's active site at any given time? So before coming to the option, let us see how it happens. Competitive binding of Rubisco. How it happens? If concentration of CO2 is greater than O2, Rubisco act as an carboxylase. That means it binds with CO2 and thus photosynthesis occurs. But if concentration of O2 is greater than CO2, Rubisco act as an oxygenase. That means it binds with O2 and thus photorespiration takes place. Student, Rubisco can bind with both CO2 as well as O2. These two gases compete for same active site. The winner is determined by their relative concentration inside the leaf tissue. More CO2 favors photosynthesis while more O2 favors photorespiration. Coming to our options. Option number 1. The absolute structural size of the chloroplast. The size of chloroplast does not determine which gas binds to the Rubisco. Binding depends on substrate availability at enzyme's active site. Therefore, this option is wrong. Option number 2. The relative concentration ratio of CO2 and O2 inside the cell tissue. Yes, this is our correct answer. Rubisco has affinity for both CO2 as well as O2, and their relative concentration determine whether the enzyme Rubisco function as carboxylase or oxygenase. Option number 3. The abundance of accessory pigments like carotenoids. Carotenoids help in light harvesting. They do not influence the competition between CO2 and O2 for Rubisco. Therefore, this is also the wrong option. Option number 4. The thickness of the outer mesophyll cell wall layer. Cell wall thickness does not directly regulate the substrate binding at the Rubisco active site. Therefore, this is also the wrong option. Moving on to MCQ number 5. Blackman's law of limiting factors 1905 governs multi-factor biological scenarios if the plant is provided with optimal light intensity and a perfect temperature but highly deficient CO2 levels, which modification will immediately accelerate the photosynthetic rate. So before coming to the options, let us see the Blackman's law of limiting factors. According to Blackman's law of limiting factors, when the several factors affect the process, the factor which is present in least favorable amount becomes the limiting factor. According to the question, light intensity is optimal and temperature is also optimal, but the concentration of CO2 is deficient. That means photosynthetic rate is limited by CO2. So student, according to Blackman's law of limiting factors, the rate of photosynthesis is controlled by the factor which is in shortest supply. That means the limiting factor. Here light and temperature is already optimal, but CO2 is deficient. Therefore, increasing CO2 concentration is the only change that will immediately increase photosynthetic rate. So coming to our options. Option number 1, increasing the ambient light intensity even further. Light is already at its optimum level. So according to Blackman's law, increasing the non-limiting factor, that is the light in this case, will not increase photosynthetic rate. Therefore, this option is wrong. Option number 2, elevating the external concentration of CO2 gas. Yes, this is our correct answer. CO2 is limiting factor. So, increasing its concentration will immediately increase photosynthetic rate. Option number 3, dropping the temperature to freezing levels to preserve enzymes. Photosynthetic enzymes require an optimum temperature range. Freezing temperature reduce enzyme activity and would lower photosynthesis. Therefore, this option is also wrong. Option number 4, spraying additional water directly onto the chlorophyll pigments. The problem is not the water availability, but insufficient CO2. Adding water does not remove the limiting factor. Therefore, this option is also wrong. Let us see MCQ number 6. Photosynthetic rate is influenced by both internal plant factors and external environmental factors. Which of the following is completely categorized as an internal plant factors? Let's see what are those factors. Factors affecting photosynthesis is divided into two groups. First is internal factors, which are plant factors, means which belongs to plant's own structure and physiology. And the second one is external factor. These are from surrounding environment. So what are those internal factors? They are leaf anatomy, and structure. Secondly, leaf orientation. Third, chlorophyll content. Then protoplasmic factors. Then accumulation of carbohydrates. Then leaf age. And what are the external factors? Light, temperature, CO₂ availability, water and humidity. Let's see our options. So question is asking which of the following is completely categorized as an internal plant factor. Option number 1, ambient CO₂ concentration and current air temperature. The word ambient and current air temperature indicate the environmental factor. These are external factors. So this option is wrong. Option number 2, the orientation of leaves, age of leaf and internal chlorophyll content. Yes, this is our right answer. Leaf age, leaf orientation and chlorophyll content are the internal plant factors. Option number 3, soil moisture content and incident sunlight angles. Soil moisture and sunlight angles, these are environmental conditions, making it external factor. Therefore, this option is also wrong. Option number 4, atmospheric humidity and wind velocity profiles. Humidity and wind velocity also belongs to external environmental factors. Therefore, this option is also wrong. Moving on to MCQ number 7. When plotting the light intensity against the rate of CO₂ fixation, the resulting curve exhibits specific regions. What does the linear straight line region at low light intensities signify? So before coming to the options, let's see our graph first. Student, this is a graph between light intensity and the photosynthetic rate, which is the rate of CO₂ fixation. And this is the light intensity. This is the first region, which is the linear region, region of low light intensity. This is the second region, which is the transition region. And the third one is saturation region, which is the region of high light intensity. Student, in this region, that is in the linear region of low light intensity, the rate of CO₂ fixation is directly proportional to the increase in light intensity. There is a linear relationship between incident light and the rate of CO₂ fixation. This means light is a limiting factor. Increasing the light directly increases photosynthesis. At low light intensity, every extra photon captured by chlorophyll directly boosts the photosynthesis. So light becomes limiting factor, and this creates a straight line relationship between light intensity and CO₂ fixation. Let's coming to our option. Option number 1, light is acting as a strong limiting factor, showing a direct proportional relationship. Yes, this is our correct answer. The straight line portion of the graph shows that light is a limiting factor, and every increase in light intensity produces a proportional increase in CO₂ fixation. Option number 2. The plant has reached maximum saturation and cannot process more light. Saturation occurs later when curves become nearly horizontal. In the linear region, the plant has not yet reached maximum photosynthetic capacity. So this option is wrong. Option number 3. Chlorophyll pigments are undergoing catastrophic photooxidation. Photooxidation is associated with excess light intensity, not the low light linear region. Therefore, this is the wrong option. Option number 4. The dark reaction enzymes have completely shut down. Calvin cycle enzymes functioning normally. In fact, as the light increases in this region, photosynthesis increases proportionally. Therefore, this option is wrong. Moving on to MCQ number 8. Light saturation for most common terrestrial plants occur at what precise percentage of full, unclouded, peak sunlight? Student, one of the most surprising fact is that most of the plants reach light saturation at only a small fraction of full sunlight. Beyond that point, extra light produces little or no increase in photosynthesis. What is light saturation concept? That full light is 100%, but most plants reach light saturation at 10%. This means more light does not mean more photosynthesis. So what is our first option? 100% of full sunlight. Plants do not need the entire intensity of full sunlight to reach the saturation. In fact, photosynthesis usually plateaus much earlier. So this option is wrong. Option number 2, 50% of full sunlight. This value is much lighter than saturation level for most plants. Therefore, this is also the wrong option. Option number 3, 10% of full sunlight. Yes, this is our correct answer. Most terrestrial plants reach light saturation at only about 10% of full unclouded sunlight. Beyond this point, increasing light intensity produces little additional increase in photosynthesis. This is why light is often not the limiting factor under natural field conditions. Option number 4, 25% of full sunlight. Although lower than 50%, it is still much higher. Therefore, it is also the wrong option. Let's see MCQ number 9. Except for plants growing in deep dense forest shade or highly crowded crop canopies, light is rarely a limiting factor in nature. What happens if a plant is exposed to extremely high incident light intensities beyond normal limits? Student, more light is beneficial only up to certain limit. Beyond that limit, excessive light begins damaging the photosynthetic machinery, especially chlorophyll pigments, and this phenomena is called photooxidation. So let's see what is photooxidation. We know that in normal light, higher photosynthesis occurs, but in case of excessive light, photooxidation occurs. So what is photooxidation? Photooxidation is light driven damage due to excessive light. So plants absorb more light energy that it can use to make food. This trapped energy has nowhere to go, so it accidentally charges O2 inside the leaf, thus creating dangerous hyperactive forms of O2, which is free radicals. These free radicals break down chlorophyll pigment and vital proteins. Thus photosynthetic rate falls. So let's see our options. Option number 1. The rate of sugar export through xylem increases exponentially. Xylem transports water and minerals, not sugars. Sugars are transported through phloem. Therefore, this is a wrong option. Option number 2. Chlorophyll molecules break down photooxidation, causing a drop in photosynthesis. Yes, this is our correct answer. Due to excessive light intensity, chlorophyll undergoes photooxidation, leading to chlorophyll breakdown and decline in photosynthetic rate. Option number 3. Rubisco mutations occur instantly to fix nitrogen gas. Rubisco does not fix nitrogen, and mutations do not occur in response to high light intensity. Therefore, this is also the wrong option. Option number 4. The light reactions shift completely into the root cells. Root cells lack chlorophyll and do not perform light reaction. Therefore, this is also the wrong option. Let's see MCQ number 10. Carbon dioxide is the major limiting factor for photosynthesis on Earth. What are the baseline ambient atmospheric CO₂ concentrations and what levels cause a stressful saturation or damage point? Student, carbon dioxide is the major limiting factor for photosynthesis because its concentration in the atmosphere is surprisingly low. Even small increase can boost photosynthesis, but only up to a certain limit. So let's see what is its concentration in the atmosphere. Atmospheric CO₂ and photosynthesis. So atmospheric CO₂ is 0.03% to 0.04%. That is equivalent to 300 to 400 ppm. So this increase in 0.05%, that means 500 ppm, causes photosynthesis to increase. But the higher levels, that is long exposure, that is more than 500 ppm, may become harmful and causing photosynthesis to decline. So let's see our options. Option number 1. 0.01% baseline, 0.02% damage point. These values are much lower than atmospheric CO₂ concentration, which we have seen that what is the atmospheric CO2 concentration? 0.03 to 0.04%. But this value is much lower than atmospheric CO2 concentration. Therefore, this is the wrong option. Option number 2, 0.03% to 0.04% baseline increases up to 0.05% can enhance rate, but higher levels become damaging. So this is our correct answer. We have already seen that atmospheric CO2 concentration is about 0.03 to 0.04%, and increasing up to 0.05% can enhance the photosynthetic rate, but higher concentration may become damaging. Now option number 3, 0.10% baseline, 0.50% damage point. These values are far above normal atmospheric concentration. So this is the wrong option. Option number 4, 1% baseline, 5% damage point. Atmospheric CO2 is nowhere near 1%. This option exaggerates the concentration by a very large margin. Therefore, this is a wrong option. With this, we have completed the photorespiration and the first two factors affecting the photosynthesis, that is light and carbon dioxide concentration. In the next video, we will discuss the remaining two factors, that is temperature and water, and understand how they influence the rate of photosynthesis. Thank you for watching.